Sorry for that bit of a delay, but I'm delighted to be here in Russia for the first time and able to speak to this wonderful meeting. I'm impressed at how it has progressed in the last 5 years and glad to be a part of it today. And I'm also glad that Alex got us set up beginning to talk about invasive approaches, which is what I'll continue talking, and in particular moving from ECOG kinds of signals to intercortical single neuron signals. My lab has developed a BMI, or BCI, in which instead of controlling the movement of a computer cursor or a robotic arm, we're actually moving a monkey's own arm, which we've temporarily paralyzed. And so it's something of a unique BCI that would be appropriate for a spinal cord injured patient. We've developed this over the last couple of years, and now we're beginning to move this system from something that would be only accessible in a very specialized laboratory environment to, we hope, something that would be more like a patient's home. So that'll be the theme I'll tell you about. The way in which we move the monkey's arm, or open and close his hand, is by using what we call functional electrical stimulation. This is something that's been used clinically for spinal cord injured patients for decades. And this first clip that I'm going to show you is a patient with an injury at about C5, which means he's unable to move, open and close his hand, but he can move his shoulder. And if you watch what he's doing, as he lifts his shoulder, it opens and closes his hand. And this is possible because there's a sensor on his shoulder which senses his residual movement that he's still capable of doing and translates that into a pattern of stimulation of 12 different muscles in his forearm and hand. So when he shrugs his shoulder, it closes his hand. When he drops his shoulder, it opens his hand. And this is a pattern of stimulation that an occupational therapist has to program. It's entirely pre-programmed, but it allows him and this other patient on the right to have significantly improved motor behaviors. So you can see here on the right is after he's implanted with this prosthesis, he's able to pick up these blocks and move them back and forth much more successfully than before. And if you watch closely, you can see him shrugging his shoulder. Now he's writing with a pen. Shrugging his shoulder to grasp the pen, and then moving his hand with the remaining natural motor activity that he has. So this kind of prosthesis, called the freehand prosthesis, has been implanted in about 250 people at the Case Western Reserve FES Center over the last couple decades. About 95% of these patients have significantly improved motor function. But you can imagine the cognitive burden that is imposed on these patients because they're literally using the same muscles of their shoulder both to position their hand in space and to open and close their hand. So it takes a significant amount of attention and learning to use this effectively. A recent survey suggested that among this population of patients with tetraplegia, 78% of them would undergo brain surgery if it would provide them with significantly improved hand function. So that's the kind of thing we're trying to accomplish. I told you that we're using single neuron recordings. Many of you are familiar with the Utah array shown in the upper left here. This is a 10 by 10 grid of electrodes. Each of them is 1.5 millimeters long. And at the very bottom on the left, you can see an interoperative picture in which we were implanting, in fact, 2 of these electrodes in a monkey. The tips of these electrodes are designed to go down to layer 5 and record the output signals from the cortex. And it's those signals that we use for our BCI. For those of you who perhaps haven't seen a monkey doing this kind of research before, this clip on the right, no audio. Imagine there are bursts of activity every time the monkey reaches with his hand. These should have been single neuron recordings, and you can hear the activity of one neuron in this monkey's brain as he reaches and grasps this food. And it's this burst of activity related to the monkey's behavior that we can record and analyze and use as our control signals. We're recording from the hand area of the cortex, so unlike these patients, they can imagine or attempt using their hands in order to open and close their hand. And this is essentially the way it works. On the left is a monkey that we train to pick up this ball and drop it into the tube at the top, and it falls out the bottom and drops it in again. And we actually designed this task to be something like the box and blocks task that the patient you saw was doing. It's a standard method for rehabilitation, evaluation, and for training of patients, and so we decided to imitate it. On the upper right is shown a raster of spike firing rates. There were about 70 simultaneously recorded neurons in this particular case, and the bright parts of these rasters are when these neurons are firing, like you would have heard in the previous clip. And it coincides with the monkey grasping this ball. In this particular figure, he's grasping 3 different times, and you can see the 3 stripes, primary stripes through the neural recordings, and also below it you can see EMG recordings that we were recording from a normal monkey as he was making these movements. And you can see there's a correspondence in time between the neural activity and the muscle activity that we recorded. And so we take advantage of this fact by essentially calculating a big multiple linear regression problem between the firing rate of these 70 neurons, and we look backwards in time, 10, 50 millisecond bins for each of the neurons. This gives us about 700 regressors that we use in order to fit each of these EMG signals. So once we've computed this regression problem, then we collect new data from the brain and from the muscles, and this transformation, this multiple regression set of equations that we've calculated allows us to make predictions of muscle activity by looking at the brain. And that's what's shown by the blue traces here. So these are predictions of muscle activity that we make by looking at the brain, and we can compare them with what we actually recorded from the muscles at the same time. And you see they match quite well. We computed R-squared between the actual and the predicted signals, and they range between something like 50%, and I forget what the highest is and I can't read it, but 85% we can typically achieve. So you see these patterns are fairly complex, and yet we can predict them quite accurately. So we can take these real-time predictions of EMG and use them to control the stimulation of the muscles, as you saw in the example with the patient, except instead of being pre-programmed, the patterns of stimulation that we deliver are determined in real time by looking at the brain. This is the way the experiment works. As I've told you, we acquire data from the monkey's brain, we transform it into muscle predictions, and then another simple transformation into the pulse width of the electrical stimulation that we deliver to each of these muscles. Now, in order to make this a reasonable model of spinal cord injury, we inject a local anesthetic into the motor nerves of the monkey's arm to temporarily paralyze his hand. So it's a bit like when you fall asleep on your arm and you wake up and you can't move it in the morning. The paralysis lasts for a minute or so. When we put in the drugs, the paralysis lasts for an hour or 2. During that period of time, we can do this experiment. And this is a clip showing exactly one of these experiments. So this monkey has a peripheral nerve block of the median and the ulnar nerves, which are the nerves that control the flexion of the wrist and the flexion of all the fingers. The first couple of trials I'll show you here are during this nerve block without the BCI running. And you can see the monkey reaches out and he tries to grasp this ball, but he can't use his hand properly. Presumably, the neurons in his brain are modulating as they normally would. The signals simply can't get to the muscles, which is essentially what happens to a spinal cord injured patient. The next trial now, we turn this prosthesis on, and the monkey can immediately use his hand voluntarily. So these hand-related signals in the brain are being processed by our computer, and in tens of milliseconds are used to stimulate the muscles and provide the monkey voluntary control of his hand. Okay? But you can see the monkey sitting here in this chair in the lab. I told you it lasts for 1 or 2 hours. And what we'd like to do is move this technology from this lab setting into the real world, or as real a world as we can do with our monkeys. In order to do that, we use commercially available wireless recording systems from BlackRock to record from the brain. We also have a wireless means of recording from muscles or stimulating those muscles while the monkey is in his home cage. So we can essentially do the experiment that I just told you about in the cage as well as in the lab. In order to keep the hand paralyzed for a longer period of time, we've replaced our literal syringe to do the peripheral nerve block with an implanted infusion pump. And this infuses the drug slowly and continuously to the nerve to keep it blocked and keep the monkey's hand paralyzed. We developed this initially in rats, and we kept a rat's sciatic nerve blocked for a month, and then turned off the pump, and his motor function returned by the next day. We did the same thing in a monkey. We kept that paralysis for just 2 weeks, but motor function returned normally after that. So this is our model of spinal cord injury, and the model that we will use to attempt to do this kind of model continuously in the monkey's cage, which we've not yet accomplished. But this is the monkey in his cage. He's wearing all of the wireless equipment, and you can see him walking around. And what we'd like to do is have him engage in the variety of things he normally does. So here he's locomoting, using his hands to grasp the rod. Now he'll bend down and pick up a tube. This tube is full of paper that you can see shredded on the ground already, and food treats inside, so he forages in that tube. He's also trained to push buttons, so there's a wide variety of different kinds of motor behaviors that this monkey can do in the cage, and we can study the activity within the brain underlying those activities, and then eventually we'll try to restore his ability to do each of these behaviors with cortically controlled FES. On the way there, we're beginning to try to understand the different behaviors that the monkey performs in the cage and the cortical activity that accompanies them. This is work done by postdoc Xuan Ma. He's looking at 16 different EMGs that we record wirelessly from a variety of muscles of the hand and arm, and essentially assembles these multiple EMG signals into a single long vector and applies a dimensionality reduction method to it. And that picture is shown here. So this is TISNI, for those of you who are familiar with it. It's a nonlinear dimensionality reduction method. And after doing TISNI, he has an automated way of coloring the different features here. It's not quite a cluster analysis, but it's essentially like a cluster analysis, with each of the clusters corresponding to patterns of EMG that were similar. Then he can go in and look at these different kinds of patterns and see what behavior they correspond to. So you can see he's labeled button pressing behavior that I showed you, and the foraging behavior, and also when the monkey's just sitting there doing nothing, with some characteristic kinds of EMG there. And it's the case that if the monkey is foraging for a while and then goes back and does something else and then returns to foraging, the EMG patterns return to the same location within this low-dimensional projection of EMG space. So he does capture something about the patterns of EMG that underlie these different kinds of behavior. So then the question is, what is the relationship between brain activity and each of these patterns of activity? We know that in the cage, if the monkey does just simple grasping tasks, we can decode it quite well. But what if the monkey does grasping for a while, foraging for a while, button pressing for a while? Can we use the same decoder to accomplish that? And the answer is, if we use either a linear decoder or a fairly simple RNN, no. This is a confusion matrix showing our ability to decode these different EMGs during these different kinds of behaviors. Just the two behaviors and the idle behavior. And you can see the diagonal is darker red than the others, which means a decoder trained on one activity predicts that activity pretty well, but it doesn't predict the other activities terribly well. Now, we've had recent success that I can't tell you about yet with a much more complicated RNN decoder, which does seem to generalize much more generally across this range of cage behavior, and we're continuing to develop that decoder. But I want to tell you about a different problem that we're also addressing with another kind of deep neural network, and that is the fact that, as someone alluded to in the morning, the Utah arrays record very well for a number of months, and then they begin to record fewer and fewer neurons. We're actually able to record often for a couple of years from an array, but it is certainly the case that the neurons we record change. So if we compute a decoder on one day and continue to use that same decoder, despite the fact that neurons are changing here with a couple of weeks or 3 weeks between them, our ability to predict declines dramatically. So if we recompute that decoder every day, that's the gray dots at the top, and the information content remains essentially constant across that period. But if we fix the decoder on day 0 and continue to use it, our ability to decode basically drops off to 0. So here we'd have a monkey trying to use this decoder with absolutely no useful information at its output. Clearly not a desirable outcome. So we're trying to address that with a deep neural network. And the basic idea of it here is shown here. I hope many of you in the room recognize that first picture on the left. This is from the work of Nikolai Bernstein in the middle of the previous century, done in Moscow. He was very interested in how the brain controls relatively simple movement with this really complicated effector. There are 27 degrees of freedom in the hand, there are another 7 in the arm, and yet we can control them very fluidly. And it turns out if you do principal component analysis or other kinds of dimensionality reductions methods like this, the dimension of activities like the one you see hammering can be effected rather well with 3 or 4 or maybe 5 degrees of freedom. So the dimensionality of the behavior is actually rather small. They're being controlled by at least 1 million neurons in the cortex, and we stick an array in that cortex and we can record about 100 neurons. So many of you are familiar, I think, with the concept of a latent space within the brain. So if I stick my electrode in the brain and record 100 neurons, I can think about that as a 100-dimensional system, if each axis represents the firing rate of one neuron. I told you the behavior is maybe order 5. So if we use methods to reduce the dimensionality, we can find a latent space, a space of signals that are about 5 to 6 dimensional that we can use to reconstruct behavior quite well. So if you think about that for a moment, we're essentially taking a random sample of neurons from the brain and we can reconstruct behavior. So really any random sample ought to be adequate to do this. So if the neurons change, it shouldn't matter. The problem is, because these latent signals are embedded in a 100-dimensional space, the axes of which keep changing, our latent space will need to be realigned on day k so that it's the same as it was on day 0. And that's essentially what we're going to try to do. And then we'll do our decoding from those latent signals instead of from the electrode signals themselves. And this is an example of how this works. We're using autoencoders to do this dimensionality reduction, but there are many different ways of doing that. An autoencoder is simply a deep network that has as inputs the neural firing rates on the top here, and its job is simply to reconstruct them, as you can see on the bottom here. The trick is the neural network has a bottleneck in the middle of it, and we can make that bottleneck as small as we want, and that causes the dimensionality reduction. So here we had a latent space of 16 dimension in the middle. And Ali used the signals from that latent space to make predictions of EMG, much like I showed you before. And you can see they match up quite well. So can we take these latent signals and realign them on successive experimental sessions such that we can continue to make good predictions? So this is a 3-dimensional plot of the latent signals that result when a monkey does what we call a center-out motion. He's making movements, reaching movements to 8 different targets, and these are the 3 first principal components in this latent space. And you can see their colors are set by the direction of reaching movements, and you can see there's an orderly progression as he moves from one target to the next target in the latent space of these neural signals. That was on day 1. If we do the same thing on day 32, you see these latent signals are completely messed up because we're recording a different set of neurons. I'm not going to go into the details here because I don't have time, but we used something called canonical correlations to line up the latent signals on day 32 with the ones we recorded on day 1. This requires that the monkey makes movements of exactly the same kinematics so that we can line up the kinematics and then line up the neural space. When we do that, we can get these kinds of trajectories. So on the left is the newly aligned signals from day 1, and on the right is the aligned signals from day 32. And you see they match quite well. We've managed to realign these 2 latent spaces so they basically look alike. If we do the decoding now from these latent signals, we get the blue dots. You see we're able, with a fixed decoder computed on day 1, that's getting input from these realigned latent signals, we can continue to decode for a month and a half with the same decoder, whereas by recomputing the decoder every day, the information goes down to 0. These were the results from one monkey. This is the result from another monkey. This is 2 years. We're able to maintain consistent decoding over a 2-year time span by realigning this latent space. However, we couldn't do the same thing with cage data because this system, this method with the canonical correlations requires that the kinematics themselves be aligned in time so that we can thus align the latent signals. So we also are working on methods that allow us not to align the latent signals themselves, but in fact the probability distribution of signals within the latent space. So there's no time here. This is simply a projection into 2 dimensions, much like the one I showed you for EMG earlier. You see there are 8 arms on this distribution of points in this neural cloud. These correspond to those 8 directions of motion that I showed you before, but time is not represented implicitly here at all. So on the top is the distribution at day 0. At the bottom is what this distribution looked like 16 days later. It doesn't match at all. Now I'm going to go through this very quickly because I'm out of time. But this is the deep neural network that we used, which essentially is a version of a GANs, Generative Adversarial Network. These are the things that are used to produce fake pictures and fake movies, in fact also to do speech recognition. And what we're using it for is to make the distribution of points on day k look like the distribution on day 0. And on the upper right you can see how successful it is. We've managed to transform this subsequent distribution of points to basically look like the distribution on the first day. And if you look at our ability to decode, the brown line and the orange line second from the top shows that we can continue to decode movement successfully across this 2-week period by using these realigned latent signals. A couple of challenges that remain are to improve our generalization, our ability to predict the signals within the cage, and then to use this alignment method on cage data as well. So to summarize, we can use cortical activity to predict EMG and to control FES stimulation intensity. There's a progressive change in neurons that causes fixed decoders to fail, but we can compensate for this turnover by using one of a couple of different methods. And these are the people that contributed to the work and the funding agencies. Thanks.
Thank you very much for the presentation. So we have time for questions. We already have one question here.
Okay. Thank you for your interesting talk. And a question may be related to a future of this technology. How do you expect to train this system on real patient when you transfer from monkeys because you don't have any valid muscle activations? What are your thoughts on this?
Yeah.
How do you plan to do this?
That's actually a great question, and it's an obvious problem we need to deal with. The way we can do it is much like what people call an observation-based decoder that many labs are using for training decoders of kinematics. Essentially, you have a patient or a monkey track a known trajectory and then compute the mapping from the brain activity that you record to the trajectory that you ask them to produce. We can't do the same thing because we have patterns of EMG, but if we have the monkey make relatively simple movements like the ones I showed you, we know that the pattern of EMG that's required for that is pretty similar even across monkeys. So we can take standard templates of EMG activity during these particular kinds of movements and use them like the observation decoders to compute a mapping from the brain activity we record while the monkey tries to do that, or a patient would try to do it, and the known EMG templates.
Oh, okay, thank you.
Vadim, another question.
Thanks. To what extent, like I understood these electrodes go to layer 5, right?
More or less.
So have you tried to also decode it with LFPs? Like,
I have a paper maybe 10 years ago with a postdoc of mine by the name of Mark Slutsky, who's now a faculty member in neurology. And we compared LFP and single units in rats, doing a simpler task. And I told him it would never work, but it was fairly successful at decoding EMGs for 4 movement directions with a little joystick, but I've not done it in monkeys.
So the reason why I ask is that if it's not only layer 5, and let us imagine that you have a perfect classifier. So how would we know that this is exactly the pattern brain uses for control, you know? This is not the same thing.
I wouldn't argue that it certainly is layer 5, and I don't think it needs to be, because the movement-related information is in most of the layers of cortex.
It goes to the question that, for instance, when people from V1 or V2 decode visual information, sometimes in V1 and V2 there are not strong horizontal connections, so it means you can decode something quite well, but neurophysiologically it's not very meaningful because brain doesn't have information about this connectivity. But it's not so applicable here because you directly have output neurons, basically.
That's right, or we're very close to them. We've actually looked at the relationship between PMD neurons and M1 during motor learning. And there are some fascinating differences between them. That's perhaps more analogous to V1 and V2.
Nice, thank you.
Thank you. One last question before the break.
So thank you. The question, I guess I will follow this topic. So, as I understand, you're hitting 100 neurons, and you hope that you hit some pattern completion neurons of some neuron ensembles which control this movement. So we have some pattern, we have ensemble of neurons which control it, and you hope that we are recording from the most important neurons of this ensemble. So the question is, but it's only 100, and this ensemble can be spatially very distributed. So the question is, how do you think your thoughts on how much neurons and on what spatial distributions you need to reliably hit all these neurons? Like you need 100 on 1 millimeter, 1,000, 3 millimeters, so to be sure that you will hit them.
Well, I don't think we can be sure, but 100 seems to be adequate to make a very reliable sample. When I began this work about 12, 13 years ago, we only had a 32-channel system, and so we had to pick, but we put in 100-channel electrode array, so we picked 32 out of the 100 that had the most information, and that worked fine as well. So while it would be great to have more and more neurons, and I think it will improve it, it's remarkable how few we actually need. But this raises one of the very interesting questions we're trying to look at in the cage. As the monkey does more and more random kinds of spontaneous behavior, we're hoping that we find the signals become more complicated, and we do need more neurons in order to represent them adequately.